During the beginning of this course, we came across work of Alvin Stackman, who in 1915, while working on cereal rust, first time used term hypersensitive response or HR. He defined it hypersensitivity of plant towards rust fungi. He noted that the area showing HR became dead. He also found a correlation exists between HR and resistance and those varieties which showed HR are resistant one, while those which showed no HR were susceptible one. HR is quite regularly used in the plant pathology laboratories all over the world, either to assess the resistance of the plant variety or to assess the pathogenicity of a microbe. Here you are seeing a plant pathology student injecting a unknown bacterium inside the tobacco plant to see whether it produced the HR or not. And on the right side you categorically can see a dead necrotic brown colored spot appear where the bacteria is injected, while the portion of the plant which is injected with the sterilized water remain green. So it means that the bacterium injected here are plant pathogenic, and secondly, the tobacco plant is either resistant or non-host to it. But the important thing is also here to note that the HR is confined to a certain area. That is why HR is also known as localized acquired resistant. Why we use the acquired? Because this resistance is due to the injection of the microbe. If the microbe is not injected, then the plant would not exhibit any kind of resistance. Frank Ross was a teacher and a researcher in the Cornell University, and after the 36 years of Stackman discovery of HR, he performed an experiment on the tobacco mosaic virus in order to see the impact of HR on the overall resistance of the plant. So he used another term which is known as systemic acquired resistance, which we are going to see how it works. I try to elaborate the work of Frank Ross by this simple picture. On the left side, you are seeing a tobacco plant which has many leaves 1, 2, 3, 4, 5. And when this tobacco plant was inoculated with a tobacco mosaic virus, and if you look at that, I gave it a black color. So what happened that this tobacco plant showed the disease symptoms which are the appearance of mosaic area. It means that the strain which is used in the inoculation was the virulent one, and the tobacco plant was susceptible against it. But in another plant, the Frank Ross changed his strategy. This time he inoculated the lower leaf of the tobacco plant with another viral tobacco mosaic virus strain, and I gave it a green color. So it is again tobacco mosaic virus, but it is different from the first one. What happened next was the production of HR. And from the Stackman work, we know that only those varieties are resistant one which gave HR. So it also means that this strain of the tobacco plant, which is shown written in the green letter, is the avirulent one, and the tobacco plant against it is the resistant one. That is why it is producing HR. But interestingly, what happened next? That on the same plant, he inoculated the strain which is the virulent one and against which this tobacco plant is susceptible one. But this time, the disease is not occurred. It means that now this plant is not only resistant against the avirulent strain, but the virulent strain of the same virus. And he repeated this experiment by first inoculating the avirulent strain to the upper leaves and inoculating the lower leaves with the virulent strain, but he got the same result, that every time the tobacco plant not show resistance against the avirulent strain but also the virulent one. So from that he inferred that this resistance which is activated in this tobacco plant is actually somehow related with the localized acquired resistance or HR, which not only confined the pathogen but also make this plant systemically resistant against the virulent pathogen. And he used the term systemic acquired resistance. And interestingly, he also noted that this resistance remain viable not for 1, 2, or 3 days, but up to 20 days. It means that if you initially inoculate the avirulent strain, the plant remain resistant against the virulent strain not for 1, 2 days, but for the 20 days. Another finding of the Frank Ross was that this resistance is not only effective against the same strain of the tobacco mosaic virus, but this plant is also become resistant to other strain of the viruses as well as against the pathogenic fungi. So the systemic acquired resistance is not only effective against the same strain, but it is also give a generalized kind of resistance, and plant once the systemic acquired resistance activated in it remain resistant against couple of pathogens. So because this resistance is not only confined to a specific area, like in case of HR, they use the or he used the term systemic acquired resistance. So from that one, I think every one of you now understand what is the systemic acquired resistance. Secondly, if you little bit compare it with the vaccination which is going on in the animals and the humans. In case of vaccination, we were given some kind of polio vaccine in our childhood, or the vaccines of the measles or the TB, and our body remained resistant for the rest of our life. So here the concept is almost same, but we not use the word vaccination. Why? Because the plants don't have antigen antibody reaction, and secondly the plant defense system is not circulative. It don't has any kind of T cells, white blood cells, or the memory cells. So in plants we use the word systemic acquired resistance. But if the similar kind of phenomena, if it occurs in the animals and the human, then the word vaccination is used. So if we want to define what is the systemic acquired resistance, it is the systematically activated resistance after primary infection with the necrotizing pathogen. This resistance is different from the non-host resistance as it is activated by some avirulent pathogen. It produces the symptom, but most of the time it is not observable in the field. Its symptom in the form of HR, but it is so minute and tiny, but we can hardly see it in the field, and that is why we don't realize that the plant are exhibiting systemic acquired resistance while they are growing in the field. But every plant exhibits systemic acquired resistance. That is why it remain viable and live despite the presence of so many virulent pathogens in the field. After the Frank Ross paper on the systemic acquired resistance, which was published in 1961, everyone in the plant pathology community was convinced about the systemic acquired resistance and totally agreed what Ross found in his research. But the one question remained pending. What was the nature of the signal which was produced at the localized acquired resistance at the HR area, and then it travel all over the plant and make this plant systemically resistant even against the virulent pathogen? You don't need to be genius to find this signal molecule. This signal molecule must be present where there is HR is going on. And if it travel down all over the plant, it must be present in the vascular tissue, but it shouldn't be present in the xylem tissue because the xylem tissue is made to take the water from the soil and then distribute it all over the plant. It means that the signaling molecule not only present at the area where the HR is going on, but it should also be present in the flowing part of the vascular tissue. And thirdly, this signaling molecule must be present where the systemic acquired resistance is exhibited against the virulent pathogen. So, experiment after experiment, the people find out that the signaling molecule which is present at HR in the phloem tissue of the phloem part of the vascular tissue, as well as in the area where the finally SR is exhibiting is the salicylic acid. So, people are convinced that the systemic acquired resistance is due to a signal molecule which is known as a salicylic acid. Salicylic acid is the plant hormone which not only has significance in plant health, but it is also very important for the human health, and it is one of the medicine which is listed in WHO. We all used Disprin whenever we have headache, and the Disprin is basically acetyl salicylic acid. Later on, the importance of salicylic acid in the systemic acquired resistance also came from the different experiment, especially from the loss of function mutant, which unable to produce the salicylic acid. And that mutant which unable to produce the salicylic acid also unable to show any kind of systemic acquired resistance. Similarly, the plants which are transformed with Naji gene, and Naji gene produces salicylate hydroxylase which degrades salicylic acid, and such plants which are transformed with Naji gene also unable to show the systemic acquired resistance. And this sportive evidence is enough to believe that the systemic acquired resistance is not without salicylic acid, and salicylic acid is actually the signaling molecule on which systemic acquired resistance is based. Till now, many plant pathologists believe that the salicylic acid is the signaling molecule in the systemic acquired resistance, without which systemic acquired resistance is not possible. The second part of this belief is okay, that without the salicylic acid, systemic acquired resistance is not possible, as we already seen in case of loss of function mutant and the plants which are transformed with the Naji gene. But the first part, that the salicylic acid is the signaling molecule in the systemic acquired resistance, is proven wrong with an excellent paper which we are going to discuss in the next slide. In 1994, almost 33 years of Frank Ross discovery of systemic acquired resistance, a paper was published, and through this paper, the authors proved that the salicylic acid actually not the signaling molecule in the systemic acquired resistance. So I want you people pay attention to understand how they prove that salicylic acid is not the signaling molecule in the systemic acquired resistance. They took two tobacco plants. One plant where you can see the X word mean that this is the tobacco variety which is known as Xanthi, and then there is an other tobacco plant where you are seeing the word N. N stand that this tobacco plant is transformed with the Naji gene. We already know that those plants which are transformed with the Naji gene unable to show any kind of systemic acquired resistance. Why? Because the Naji gene actually degrade produce the enzyme which degrade the salicylic acid. So first they actually perform a grafting experiment where they use the sign stalk and the root stalk. So first they want to know whether their grafting work or not. In this case, they grafted Xanthi plant on the rootstock, which is also Xanthi. And then they inoculated the rootstock with the avirulent strain, which produce HR. And then after that, they inoculated the same plant with the virulent strain, and this plant exhibited SAR. It means that their grafting works. In the second case, they grafted the Naji plant on a rootstock, which is also Naji gene. And at the rootstock, first they inoculated the tobacco mosaic virus. There is no production of HR both in the rootstock as well as in the sign stalk, which means that their grafting also work in case of Naji plants, and they don't allow the salicylic acid production, and as a result of that there is no SAR. In the third experiment, they changed their strategy and they used Naji plant as a sign stock and the Xanthi plant as the rootstock. First, they inoculated the Xanthi plant with the avirulent strain of tobacco mosaic virus. So, if it is a virulent strain, the Xanthi plant produce HR against it. It means that the salicylic acid is produced, and this salicylic acid traveled through the phloem to the upper part of the plant. And when they inoculated this Naji sign stalk with the virulent strain, there is no production of the systemic acquired resistance. But it makes sense. Why? Because despite the salicylic acid is produced, despite it traveled, but because it is the plant is challenged, is the transform with the Naji gene, and that plant degraded the salicylic acid, so there is no production of the systemic acquired resistance. In the last part of their experiment, they used Naji plant as the rootstock while the Xanthi plant as the sign stock. And we all know that in the Naji plant there is no production of salicylic acid, while in the Xanthi plant there is the production of salicylic acid is possible. So what they did? So I again say, please put little attention over here. So they inoculated the Naji rootstock with the tobacco mosaic virus. There is no production of HR because there is no production of salicylic acid, because the Naji plant carries the enzyme which degrade the salicylic acid. But interestingly, rather astonishingly, when they inoculated the virulent strain on the sign stock, which is a xanthi variety, the SAR produce. It means that there is SR can be produced uh, without salicylic acid act as a signaling molecule. Why? Because if the salicylic acid is the signaling molecule, it is degraded over there. And if it is degraded over there, then it should not be present in the phloem and it should not be traveled to the sign stalk. But the sign stalk still exhibiting the SAR. So from this experiment, they proved that the salicylic acid is necessary for the systemic acquired resistance or for the induction of the systemic acquired resistance, but that salicylic acid is not the signaling molecule. So I again emphasize to put more attention to this part of the talk. And if still you not understand, you can ask me the question about it. So, from this experiment, the authors concluded that salicylic acid was not a signal molecule because if rootstock carried Nodgy gene, it meant that salicylic acid was degraded, but still, when sign stock xanthi inoculated SAR was produced. Despite it is proven in 1994, but still you can find in many books where the authors claimed that the salicylic acid is a signaling molecule in systemic acquired resistance, which is wrong. Salicylic acid is just necessary for the induction of systemic acquired resistance, but it is never the signaling molecule in systemic acquired resistance. Systemic acquired resistance is one of the key area in plant pathology, and people using it as a tool to induce resistance well before the attack of virulent biotrophs. And the second thing, the people or the farmers also applying salicylic acid to induce systemic acquired resistance in plants because the application of salicylic acid is easy, and the second, salicylic acid is very cheap and the safe chemical. But as a plant pathologist, we must always remember that the systemic acquired resistance is only effective against the biotrophs or the hemibiotrophs, means these are those pathogens which live in a living host, but it is never effective against the necrotrophs, those pathogens which first kill their host and then live on it. And if you apply it against the necrotrophs, it will help them and there would be more disease. So at this point, I would like to finish my first part of talk on the systemic acquired resistance. And I hope that all of you have a basic idea what is the systemic acquired resistance and how it induce and how it works. Have a nice day. Goodbye.